Bioremediation of oil spills. Review of literature. The topic is Bioremediation of oil spills. Taken from the context, Bioremediation of oil spills, research paper by Sir Macaulay and Deborah Rees, accepted on March 5, 2014. The study reviews the challenges in managing oil spills in terrestrial and marine environment to identify areas that require future research. The use of encapsulating agents and genetically engineered microbes may increase the efficiency in field, but temporarily indigenous oleophilic microbes may be employed in the field. Introduction. Bioremediation employs the use of living organisms like microbes and bacteria in the removal of contaminants, pollutants, and toxic from soil, water, and other environment. Remediation of contaminated environment is a process in which the biological pathways within microorganisms or plants are used to degrade or sequester toxic hydrocarbons, heavy metals, and other volatile organic compounds found within fossil fuels. Remediation could be done ex situ or in situ through various technologies. The ex situ method involves removal of the contaminated soil or water to clean up on another surface. Oil spills that happen in rivers, bays, and the ocean, mostly caused by accidents involving tankers, pipelines, refineries, drilling rigs, and storage facilities, recreational boats, and in marinas. Oil consists of many different toxic compounds. These toxic compounds can cause several health problems like heart damage, stunted growth, immune system effects, and even death. Oil destroys the insulating ability of fur bearing mammals such as sea otters and the water repellency of birds' feather. Significance of the study. Bioremediation helps clean up polluted environments including soils, groundwater and marine environments such system can include bacteria, fungi, algae and plant species. They are capable of metabolizing, immobilizing or absorbing toxic compounds from their environment. Aim and objectives. To study about bioremediation of oil spills. Objectives: to study about spilled petroleum hydrocarbons, to study about crude oil bioremediation strategies, to study about various microorganisms involved in bioremediation of oil spills. Statement of problem. First, oleophilic microbes. Oleophilic microbes can be supported in breaking down recalcitrant asphaltenes both on land and water. Second, algal blooms. Increased quantity of algal blooms can lead to eutrophication, which has adverse effect on the environment into the water. Unsustainability of bioaugmentation in the field. Poor availability of spilled petroleum. Fifth, inefficiency of biodegradation in anoxic environment. Research work method. Bioremediation is a research intensive technology that requires a prior thorough understanding of microbial processes. Otherwise, the consequences would be more serious than the original contaminant to the ecosystem. The microorganisms act well only when the waste matter help them to generate energy and nutrients to build up more cells. When the native microorganism lacks biodegradation capacity, genetically engineered microorganisms, or also known as GEMs, may be added to the surface during in situ bioremediation. But stimulation of indigenous microorganisms is preferred over addition of GEMs. The intrinsic, that is, inherent capacity of microorganisms to metabolize the contaminants should be tested at laboratory and field levels. Through site monitoring programs, progress of bioremediation should be recorded time to time. Other environmental factors such as pH, concentration, temperature, and nutrient availability determines whether or not bioremediation takes place. Scope and limitations. First, Understanding how oleophilic microbes can be supported in breaking down recalcitrant asphaltenes both on land and water. Second, investigating how the delay in heavy pH biodegradation can be reduced. Third, the methods that can be employed to reduce the effect of eutrophication caused by biostimulation. Fourth, how the problem of bioavailability of liquid hydrocarbon both on land and water can be addressed. Fifth, reasons why experiments rather than laboratory based experiments need to be encouraged. Discussion. First, microbial consortia have more of a promising potential to break down asphaltenes than microbial isolates. Therefore, there is a need to carry out further research into identifying the functions of the individual microbes in the asphaltene degrading consortium and finding out if new microbes can be added to facilitate greater asphaltene biodegradation. Second, the possibility of harvesting indigenous microbes from the contaminated site, culturing and reintroducing them to the site may probably solve the problems of environmental intolerance by exogenous microbes. Third, the multi-component microbial consortium selected for bioaugmentation should not have antagonistic oleophilic microbes such as thalassolitas as part of the consortium, as these may affect the potency of the approach. Conclusion. The challenges associated with the bioremediation of oil spills have been investigated. The recent strategies developed to meet the challenges are also generally limited by one or more of the following: inadequate field trials in case of encapsulation, HGT, biosurfactants, and oleophilic anaerobes, stringent regulation, and negative public perceptions in the case of gem application, high cost of production in the case of oleophilic nutrient and biosurfactant production. Therefore, when the both government and governmental institutions sponsor field studies, it will enable researchers to compare the efficiency of the existing bioremediation technologies and devise eco-friendly ways in which they can be improved within minimum cost. Bioremediation is an emerging technology which can be simultaneously used with other physical and chemical treatment methods for complete management of diverse group of environmental pollutants. Bioremediation has become an accepted remediation technology and is continually evolving. This picture here shows the physical removal of oil from the ocean body by workers. These are the materials that we use for our study. This is the bibliography of the paper. Thank you very much.